Chapter 3, Nanoelectronic Lab-on-Chip Systems. Nanoelectronic Lab-on-Chip, that is LOC systems, represent a transformative approach to miniaturizing and integrating analytical processes onto a single chip scale platform. By leveraging nanoelectronic components, microfluidics, and advanced sensing technologies, these systems enable rapid, portable, and cost effective analysis of biological, chemical, and environmental samples. In this chapter, we will explore the principles, applications, design considerations, and future prospects of nanoelectronic LOC systems. What is lab on chip system? Imagine having a fully functional laboratory condensed onto a single chip, capable of performing multiple biochemical analysis with minimal sample volume and time. Lab on chip systems are exactly that, integrating various laboratory functions into a compact device, enabling rapid and automated analysis of biological samples. These systems utilize microfluidic technology to manipulate small volumes of fluids through tiny channels and chambers on the chip. By integrating sensors, pumps, valves, and other components, LOC devices can carry out complex biochemical reactions and analysis with high precision and efficiency. The process begins when a biological sample, such as blood or saliva, is introduced into the chip through an inlet. This sample may be pre-treated to isolate specific components or mixed with reagents for further processing. Once inside, the sample is directed through microchannels where it interacts with other fluids, reagents, or nanoparticles. These interactions are meticulously controlled to carry out specific reactions or separations necessary for the diagnostic test. The core analytical processes of the lab-on-chip system take place in the designated chamber or zones. Chemical reactions such as polymerase chain reaction, that is PCR, for DNA amplification are performed within these confined spaces. Integrated sensors then detect the outcomes of these reactions. Optical sensors might measure changes in light absorption or fluorescence, while electrical sensors could track variations in conductivity or impedance. The data collected from these sensors are processed by the chip's onboard electronics, which then display or transmit the results for further analysis. This automated and precise method not only speeds up the diagnostic process but also enhances its accuracy and efficiency, making lab-on-chip systems a powerful tool in modern healthcare. Lab-on-chip systems have a wide range of applications across different fields, including environmental monitoring, food safety, and most notably, medical diagnostics. In healthcare, lab-on-chip devices have the potential to revolutionize the way diseases are diagnosed and monitored. One of the most significant advantages of lab-on-chip systems in healthcare is their ability to detect diseases at an early stage. By analyzing biological samples such as blood or urine, these devices can identify biomarkers that indicate the presence of a disease long before symptoms appear. Early detection is crucial for the effective treatment and management of diseases such as cancer, cardiovascular diseases, and infectious diseases. Additionally, lab-on-chip devices enable rapid on-site testing without the need to send samples to centralized laboratory. This capability, known as point-of-care testing, brings diagnostic testing closer to the patient, allowing for immediate diagnosis and treatment decisions. Point-of-care testing, that is POCT, is especially valuable in remote or underserved areas where access to laboratory facilities is limited. Lab-on-chip systems are instrumental in detecting infectious diseases quickly and accurately. They can identify pathogens such as bacteria or viruses by analyzing biological samples, enabling healthcare providers to initiate appropriate treatments promptly. During outbreaks or pandemics, these devices play a critical role in monitoring the spread of diseases and implementing effective public health interventions. Furthermore, the miniaturization and automation capabilities of lab on chip systems support personalized medicine approaches. By analyzing a patient's genetic makeup or specific biomarkers, these devices can tailor treatments to individual characteristics, optimizing therapeutic outcomes and minimizing side effects. For example, pharmacogenomic testing can be performed to determine how a patient will respond to a particular medication. Lab on chip devices are increasingly used in cancer diagnostics and monitoring. They can detect cancer biomarkers in blood samples, analyze tumor cells, or even isolate circulating tumor cells, that is CTCs, for further analysis. These capabilities allow for early cancer detection, monitoring of treatment efficacy, and assessment of disease progression, improving patient outcomes and survival rates. Moreover, lab on chip systems accelerate drug development by enabling high throughput screening of potential drug candidates. These devices can simulate human physiological conditions more accurately than traditional methods, providing insights into how drugs interact with cells or tissues. This allows pharmaceutical companies to identify promising drug candidates and reduce the time and cost of bringing new therapies to the market. In emergency and critical care settings, lab on chip systems provide rapid diagnostic information that is crucial for making timely treatment decisions. For example, they can analyze cardiac biomarkers to diagnose heart attacks and measure electrolytes in patients with metabolic imbalances. These devices contribute to improve patient outcomes and reduce healthcare costs by streamlining diagnostic workflows and optimizing resource allocation. Despite their many advantages, lab on chip systems face several challenges that need to be addressed to realize their full potential in medical diagnostics. Technically, achieving robust miniaturization and the integration of complex functions on a single chip remains challenging. Additionally, there is a need to improve the sensitivity and specificity of detection methods to ensure accurate diagnosis across diverse patient populations. Another significant technical hurdle is scaling up production while maintaining quality and cost effectiveness. Regulatory and adoption challenges also pose significant barriers. Meeting stringent regulatory requirements for clinical validation, safety, efficacy is a formidable task. Moreover, integrating lab on chip devices into existing healthcare systems and workflows is necessary for widespread adoption. Economic and accessibility issues further complicate the landscape. Ensuring these devices are affordable and cost effective is crucial for their widespread use. Additionally, bridging the gap in access to advanced diagnostic technologies between developed and developing regions is essential. Looking ahead, several advancements are expected to enhance the capabilities of lab on a chip systems. The use of advanced materials, such as nanomaterials and smart polymers, can significantly improve device performance and functionality. Integrating artificial intelligence and machine learning algorithms will facilitate real time data analysis and decision support. Furthermore, the development of organ on a chip models to simulate human organs holds great promise for drug testing and disease modeling. In conclusion, nanoelectronic lab on a chip systems hold immense promise for improving medical diagnosis and treatment across various healthcare settings. Their ability to perform rapid, sensitive, and specific analysis using minimal sample volumes makes them invaluable tools in the era of personalized medicine and point of care testing. As advancements continue, lab on a chip systems are poised to become integral components of future healthcare systems, enhancing patient care and outcomes worldwide.